This is an overview of an article entitled Multiplexed Targeted Resequencing Identifies Coding and Regulatory Variation Underlying Phenotypic Extremes of HDL Cholesterol in Humans, published in the July 2018 issue of Circulation, Genomic and Precision Medicine. Genome-wide association studies, or GWASs, have uncovered common variants at many loci influencing human complex traits, such as high-density lipoprotein cholesterol, or HDL-C. However, the contribution of the identified genes is difficult to ascertain from current efforts interrogating common variants with small effects. Thus, there's a pressing need for scalable, cost-effective strategies for uncovering causal variants, many of which might be rare and non-coding and have large effects on the phenotype in question. In the paper under discussion, the authors started with the hypothesis that non-coding variation at HDL-C loci could underlie extreme HDL phenotypes, similar to coding variation that's been identified by traditional targeted resequencing approaches. The authors sought to apply multiplexed targeted sequencing with the purpose of identifying novel and known non-coding variants underlying HDL-C, adapting a recently reported target capture method involving molecular inversion probes, or MIPs. They applied MIP sequencing to 7 known HDL-C loci, focusing especially on non-coding regions with regulatory potential, for example, having known enhancer marks in hepatocytes. They used an extreme phenotype study design with 797 individuals with extremely high HDL-C levels, as well as 735 individuals with low HDL-C levels to serve as control individuals. Notably, the sequencing costs for the entire project only cost about $2,000 US, underscoring the potential cost-effectiveness of MIP sequencing. The authors also used traditional exome array genotyping for these individuals. This allowed them to compare results for variants covered by both MIP sequencing and the exome array. Reassuringly, the results were highly concordant, validating the MIP sequencing approach. Here's a schematic of the MIP sequencing approach. Full details can be found in the online supplement of the published article. Essentially, each MIP can capture a predefined target genomic sequence 112 base pairs in length. The authors used more than 1,500 MIPs to generate capture libraries covering more than 50 kilobases in the desired regions in the 7 HDL-C loci. Through the use of bar-coded adapters, they were able to sequence all of the libraries simultaneously, using a single lane on a next-generation sequencing machine. Here's a schematic showing the 7 HDL-C loci, with the regions of MIP sequencing shown as dark blue bars. The authors chose 4 loci with genes well known to be involved in HDL metabolism prior to the GWAS era, as well as 3 loci subsequently discovered by GWAS, 2 of which, CCDC92 and SBN01, are very new and haven't yet been deeply explored with respect to common and rare variation. The MIP sequencing identified a total of 1,956 single nucleotide polymorphisms, or SNPs, and 689 distinct insertion or deletion events, or indels, for a total of 2,645 variants. The vast majority of these variants were rare. More than 2,000 of the variants were novel, that is, they had not been previously annotated. The figure shown here summarizes other properties of the identified variants. The authors used these variants for association analyses for elevated HDL-C levels, employing a variety of strategies, as shown in the schematic and described in detail in the published article. They performed single variant association analyses with both common variants and rare variants. They also performed burden tests, in which they aggregated various groups of rare variants in the loci. An encouraging finding was that the MIP-based single variant association analyses found significant or nominally significant associations with elevated HDL-C levels, along with matching directionality, for a majority of the variants that previously had been reported to be associated with HDL-C in GWAS studies. The analyses also found a number of novel coding variants and novel non-coding variants with HDL-C associations. Shown here are a few such variants in the CETP and LIPG loci. In conclusion, the MIP-based targeted sequencing approach demonstrated the successful capture of non-coding regions for the discovery of rare non-coding variants associated with HDL-C in a cohort of extremely high versus low HDL-C individuals. MIP sequencing thus offers a scalable and cost-effective targeted approach that could complement future, larger candidate loci resequencing efforts for the discovery of candidate causal non-coding variants. Coupled with appropriate functional investigation of identified variants for impact on gene regulation, these efforts might substantially refine the causal genes at loci implicated by GWAS studies.